So we're going to move on now to IDT Biologika. Dr. Anne Bouchet, Senior Manager of New Account Management. And Dr. Bouchet will be presenting cell expansion, viral vector manufacturing for cell and gene therapeutics and vaccines and oncolytic viruses, capabilities and capacity.
Thank you, Mark. So, good morning and good afternoon, everyone. I'm happy to present you today our CDMO, IDT Biologika. IDT Biologika is a CDMO really focused on human vaccine, cells, and gene therapy, but also on the fill and finish for biologics. So our core competency is we are leading in producing the various viral vectors we are producing for vaccine. We produce live viral vaccine, we produce inactivated vaccine. We can go up to BSL-3, subunit vaccine, virus-like particles. But we develop more and more our competencies for providing services for the cells and gene therapy through viral vectors, oncolytic virus. We are developing a platform for AAV vectors or lentivirus vectors. But also we have experience in different viruses, like we can do vaccinia, pox virus, MVA. We do a lot of adenovirus. We do measles virus, RSV, HSV. Name it. So we have all these competencies in virus productions, and we also work for biologics in mRNA and MAPS. So IDT Biologika is a German-based company as a headquarter. We just celebrated 100 years anniversary last year. So you can imagine through the 100 years, we had worked with a lot of different viruses, and we have a large significant experience in this area. And we are now more than 30 years experience in commercial manufacturing. But still we are a mid-sized CDMO with more than 1,500 employees, and we are privately owned, family-owned company. So we have two sites. On the left, on the right corner, you can see here the green, it's our sites in Dessau. Dessau is some Germany and is located southwest of Berlin, about an hour, 15 minutes driving. And here we provide a full capacity from process development to packaging, fill and finish, and for small scale as large scale. And we have also a site in Rockville, Maryland, where we provide drug substance and fill and finish for early phase clinical development, phase 1-2. Both sites, we are EMA, FDA, and VIVA approved. So, as I said, we are end-to-end services for vaccine and cells and gene therapy. We also have a department in Magdeburg for process development for drug substance. So we can do all your process development. We have a large capacity to do drug substance manufacturing. We can do manufacturing of mammalian cell line, adherent or suspension cells. We have a full flexibility of different manufacturing equipment. We can work with CS, cell stacks. We can work with, we have 2 robotters. We work with isolates. So we work with Universal, and we'll discuss this later on. And we provide you process development, transfer, optimization, and scale up. We can also work on the formulation and develop also analysis for you. We can produce you in GMP manufacturing, but also clinical manufacturing from phase 1 to phase 3, and a full commercial manufacturing. And outside the drug substance, we do fill and finish, and the fill and finish services is for all kinds of biologics, where we can do formulation, filling in vials, pre-filled syringes, and auto-injectors. We can do the development of formulation analysis. We also do process transfer, scale-up. We can work with small-scale clinical trial 2-3 to up to large-scale commercial manufacturing. And we do labeling, packaging, and sterilization for commercial. With labeling and packaging, we can do also, of course, we go for the visual inspections, and we have a large lab to do quality control. So I would like to present you now the new capacity that we are developing since 2022 and will be released this year and next year. So we have decided to increase our drug substance capacities. Today we have capacity for 6 factories, isolis, spheric tank bioreactors, but for single-use bioreactors we can go up to 6 times 200 liters, and we have roller bottles, 500 roller bottles. We will have a new building. You can see here in the central the picture of the building. This is a new drug substance building. It's about 2,400 square meters. And can be here, go up to 6 times 2,000 liter bioreactors. That will be able to produce 165 batches per year. And we have a full flexibility of the upstream and downstream process technology for adherent and suspension cell line. And we can still now, so interested, please contact us. We can customize suites made on your demand. Outside this building, we decided to upscale one of a suite from BSL2 to BSL3. This suite contains 4 isolis 500. And we can do some manufacturing of adherent cell line, of course, and this will be available for manufacturing in Q2 2023. I forgot to tell you that the new building will enable us to transfer by November 2022 and the GMP manufacturing by Q2 2023. So we have an increase in capacity of single-use bioreactors from 200 to 2,000 liters. But we didn't forget about the fixed bed bioreactors. And today we increase from 1 to 5, the isolis 500. But not only that, we decided also to move to universal fixed bed bioreactors. We can go for the Carbo, which is a 30 square meters provider, to the Nevoline, 200 and 600 square meters. So we increase the drug substance capacities. Now we're going to also increase the drug product capacities, the fill and finish. And here we're going to have really soon a new high-speed filling line, the line 2, that will go up to BSL2. It will enable the biologics, including suspensions for liquid 2R, 6R, and 10R vials. We can go batch size from 50,000 to 500,000 for 2R vials. The speed line is 32,000 vials 2R per hour. This is 100% camera inspected, and it provides a low dead volume. This filling line will be available to transfer by Q3 2022 and for the GMP manufacturing by June next year. Outside this large scale, we have also increased capacity for the small scale, the one that we need often for the gene and cell therapy. So line 4 will go up to BSL2. It's adapted to the cells and gene therapy because we can deal with 2R, 10R, CZ vials. We use ready-to-use vials. We can go small clinical batch to bigger commercial scale. We can do from 100 couples of vials to 10,000 vials and 2R vials for liquid, but we can also have a lyo, and we can go up to 3,000 vials of lyo per batch. This equipment is very adapted to very high pricing product because it has a very dead volume of few milliliters. And this completes our filling line current capacity of 11 filling lines from vials to prefilled syringes. We increase the drug substance, we increase the filling line, we need to increase the logistics, the visual inspections, and the packaging. And this is what we're doing too. So associated today in visual inspections, we have manual and semi-automated visual inspections. We have a team of more than 170 people working in inspections. And we're adding a new fully automated vision inspection, high performance. We can go to 65,000 vials an hour. This will be dedicated to large batches and commercial supply. But the advantage for gene and cell therapy is moving our product to this automated filling line. We have more capacity in manual vision inspections, where we have room from 2 people to 12 people working together. Additionally to the visual inspections, we have a new capacity in logistics. We will have new storage area. So we're going to move from 13,000 pallets capacities to 40,000 pallets capacity. We will have a new drug substance storage with multiple working chambers from -80 to -20 degrees. We will have area for freezing and storing the drug substance. It will be fully dedicated to the drug substance storage. And also we integrate completely a product flow from visual inspection, packaging, storage to match this increased capacity. And the packaging, the labeling, the second packaging for clinical and commercial will follow this increase of capacity. We can provide clinical packaging, we can add room temperature 2 to 8 degrees, and also cold chain packaging at -20 degrees and below. So I would like to thank you. And now I would like to, we're going to have the poll question. And I will let our colleague present the poll question. And after that, the movie, sorry, we're going to have the movie and after the poll question. And this movie is going to present IDT capabilities in Germany. Okay, so now we have a poll question, and I would like to ask you if you can tell us if you are looking for new drug substance manufacturing capacity and what kind. Are you interested in single-use bioreactors? Are you interested in suspension cell line, 500 liter or more? Are you interested in isolated fixed bed reactor? Are you interested in universal fixed bed bioreactors, the Carbo Nevo line? Are you interested in single-use bioreactors with microcarriers? Not looking at new capacities or unsure? Please answer. We will be pleased to come back to you.
Thank you, Anne. Appreciate it. We're now opening the floor to questions for Anne, and please send any questions you have around the presentation that you just had. And in the meantime, I'll ask one or two myself here. So Anne, you talked about a significant amount of new capacity coming online. Can you talk a little bit about the QC capacity that's going to be working in lockstep with the new overall capacities that you guys have been building out?
Yes. In fact, thank you for asking, Mark, because in fact, I didn't really talk about the QC. We have in-house, we can cover about 90% of our capacity, all the tests we need in-house in Germany. And we have about more than 240 people working in QC lab. We can do transfer and development and analytics. We have developed two technologies that really is very useful. we use the next generation sequencing, the NGS, for advantageous agent testing, which really makes easier, faster, and even cheaper the technique and the testing, because they really low down compared to the in vivo or cells culture. On the other hand, we also provide FACS. And the FACS technology is very useful. It's a fluorescence activated cell vector sorter, and the FACS enable you to do the quantification, the potency determination, the characterization of our vaccine. And really, it's very useful for many batches when you start to go commercial, for really getting faster and easier release. And it's very more precise.
Okay. Great. And are you guys looking at any additional capacity expansion beyond what you've already talked about today?
Capacity in, sorry?
Capability or capacity expansion beyond what you've talked about today in the near future?
Yes. In fact, we are now working, so we move drug substance, drug product, what I described, we're going to release this year and next year. We have all the logistic behind that's going to be released slightly later. And we are also working on developing the drug product. We're releasing the vials, and we are thinking about pre-filled syringes. And now we are on the way to define what are the need for pre-filled syringes.
Okay. So your assets that you've built out here are predominantly focused on allogeneic, I would assume, on the cell therapy space, correct?
Yeah. We are, as I said, we are a specialist in virus and viral vectors manufacturing. So we are fully dedicated. And so this is where we are developing more and more gene and cell therapy needs. So AAV platform, lentivirus platform, and the virus vectors that is necessary for oncolytic virus. This is really our focus today.
Okay, great. Thank you.
You're welcome.
Any other questions? All right, why don't we move on here? Thank you, Anne, very much for your talk today.
You're welcome. Thank you.